First generation sequencing. It developed in 1970 and 1980. This generation sequencing have two main categories. The first one is single sequencing and maximum Gilbert sequencing. So it is the complete workflow of single sequencing. And single sequencing, first step is DNA regeneration. Double-stranded DNA convert into single DNA standard string and with the help of enzymes. After that, the second step is primer annealing. A short primer binds to the template, and after that, the third step is DNA polymerase. Extend the primer. An reaction matrix that contains D NTPs. These are the normal NTPs like adenine, cytosine, guanine, thymine, and also D NTPs, deoxyribonucleotide. These are the terminators for each individual nucleotide. After that, DNA polymerase reaction started so that it become a complete DNA, and in this used of buffer and magnesium, etc. And the fourth step, a random termination occur. Whenever DD NTPs is added, the elongation stop and multiple fragments from the different length we get. And fifth step, capillary electrophoresis. Each fragment pass through a thin capillaries, laser detect fluorescent, labels the fluorescence, and also we know the DNA contains four nucleotides: adenine, guanine, cytosine, and thymine. It gets four different colors. Okay, each color determined the individual nucleotide. And sixth step, chromatogram out we get. It's the software interpret the peaks that we get from the fluorescent dye. Fluorescent as the color. Okay, that gave us like if we talk about the color of different nucleotide, A get red, for example, thymine get yellow, cytosine get blue, and guanine get green. These peaks are interpreted and software and give us final DNA sequence. What are the application of single sequencing? There are multiple applications of single sequence. First application is on the base of accuracy. Single sequence is high accurate but time and cost consuming. It's highly costly and also consume time. The accuracy rate is 1 in 1000 base, and single sequence is still used today. Forward for the recurring precision and also mutation confirmations, clinical diagnosis, plasmid sequencing, cloning verification, small viral genome, and forensic science. In these areas, single sequence is still used. So this is the graphical interpretation of single sequencing. As we discussed in the first step, that this is the denaturation of DNA using heat. Okay, we use enzymes, so we need to start the denaturation of DNA. After that, it gets multiple copies. Each copy get a primer and also a four different polymerase solution. For example, this color is for adenine, yellow is for guanine, cytosine, and so forth. And fifth step, grow complementary. Then until termination dye. What was the until termination dye? DDNTP. Okay, deoxyribonucleosides are the terminations dye. After that, again we perform denaturation of the growth chain. After that, we perform electrophoresis for the four solutions, and they get us the DNA sequence. So this is the another graphical visualization of single sequencing. So reagent DNA template. Each DNA template like adenine, DD ATPs, thymine, DD TTPs, guanine, DD GTPs, and cytosine, DD GTPs. And also it goes polymerase reaction. There are DD NTPs and primer along with the reagent. Primer start aligning and chain extension. And furthermore, DD NTPs binding with the chain terminations. If they get termination, they stop elongations. And third step, we can see fluorescently labeled DNA sample. Each fluorescence have different labeled DNA samples and also nucleotide T, C, G, A, and also capillary gel electrophoresis and fluorescence detection. On the base of this DNA sample is detected with the laser detector and also sequence analysis and reconstruction of the nucleotide on the base of their position. How they detect, we can see here GAC 1, 2, 3 up to 10. These are the nucleotides they detect like this.